Okay, go. All right, so for number one, it says to draw a diagram of an intermediate stage of DNA replication of the bacterium E. coli. And so we need to draw that, and there's a couple things we need to label. Part A says we need to label the ORIC, which is at the center on both of them, right there. The DNA primase, which is here, and that is leading the helicase along. Making the primers. The DNA helicase is right here and right here, which is separating the strands. The RNA primers for the leading and lagging strands. And so here's the leading strand. And two leading strands right here. Again, I'll do that one off. DNA polymerase 1, which is down here. Chugging along. The DNA polymerase 3 alpha subunit. DNA polymerase 3 is here. And several Ozaki fragments. So we've got a couple just along the way of the Ozaki fragments. And then the polarity, we have the 5 prime, 3 prime, and then 3 prime, 5 prime.
All right, for the next one, you kind of just have to go through and figure out kind of like the definition of each of the proteins, which is kind of difficult because you got to figure it out, and I don't think the notes were that great. Not Sister Mayo's. They were just, they're hard to follow, a little confusing to jump all over the place. But the protein complex synthesizing primers, that one's pretty easy, it's just primase. So the primase makes primers. The protein promotes the separation of AT-rich sequence within the origin, so where there's a bunch of ATs, A's and T's that have least amount of hydrogen bonding, so you can pull them apart as DNA A. The subunit of DNA polymerase 3 contains the proofreading. The epsilon subunit does that, goes along and proofreads. The epsilon subunit also synthesizes the leading strand in eukaryotes. TUS is a protein that displaces the DNA polymerase at the end. So when it's done replicating, TUS knocks it off. DNA polymerase 1 has a 5' to 3' polymerase and a 3' to 5' exonuclease. Just from the notes, topoisomerase is what prevents negative supercoiling. So, in this picture right here, I don't know if you can see it, so it doesn't wind, so it doesn't super wind. The ORC, the origin of replication, binds directly to the ARS elements. Single-stranded binding proteins prevent the DNA from coming back together, so like little balls, so the protein can't collapse back down or anneal back to itself. DNA ligase, this enzyme, DNA ligase ligates together the Ozaki fragments to the thing.
Okay. So number 3 says this question concerns the enzymes involved in DNA replication. For A, it says list 5 enzymes, prokaryotic or eukaryotic, that consume ATP during DNA replication. So this is from the notes, and it's what we learned. So for A, 5 of them are DNA helicase, topoisomerase 2, DNA primase, DNA ligase, and DNA alpha. B, or DNA A. For B, it says list 5 enzymes that consume DATP during DNA replication. Those are DNA polymerase 3, DNA polymerase 1, DNA polymerase D, DNA polymerase alpha, and DNA polymerase epsilon. For C, it says list 5 enzymes, prokaryotic or eukaryotic, that have a 3' to 5' exonuclease activity. These are DNA polymerase 3, which we can see on that picture that was there. DNA polymerase 1, also DNA polymerase D, DNA polymerase, or actually, sorry, I started reading number 2 again. So C is DNA polymerase 3, DNA polymerase 1, DNA polymerase epsilon, DNA polymerase delta, DNA polymerase gamma. D, it says indicate 1 enzyme that's involved in replication that uses DNA as a template to synthesize RNA, and that's DNA primase. That's just one of them. E is indicate 1 enzyme that uses RNA as a template to synthesize DNA, and that is DNA telomerase. So 4.
All right, so 4, I'm writing up the sequence on the board. So it says you have the sequence of a single-stranded DNA molecule. Basically, wants you to make the primers. Determine the sequence of DNA strands, and says if you use the DNA nucleotides described in A through D. So A says the nucleotides in this reaction include D ATP, D CDP, D GDP, and D TTP. And if it's got a triphosphate, that means it can synthesize. So what you have to do is you got to take this, and then it says in the problem that you have a primer. You have a primer of 5' TGC GG. What you'll do is you'll take it and figure out where on the sequence it matches up. Make sure you go 3' to 5'. So basically it matches up right here, 5' end. And it's going to be, I can just do this, T, C, T, G, oops, it's A, G, 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 G. I think it stops right there, yeah. 3'. So you know this section right here is going to be a section that's going to be copied. And then if you have all of them, that'll be the section. So for A, it's going to be this whole thing, A, T, 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 all the way through, del, A, G, T, 3', 5'. And then for the next one, if you look at it, it says you have D, ADP, D, C, NP, so you have a dinucleotide cytosine monophosphate, which means you can't put in any cytosines. So the second you run into where you're going to need a cytosine, which you're synthesizing this way because you synthesize 5' to 3'. So the second you run into it, so the first one would be A, 5', and then, oh, you're going to need a C. You can't do that, so that's it. So that's how you do it for B, and then C and D. You do the same thing. You just got to figure out which one would not be able to synthesize. So in C, the UTP, uracil triphosphate, can't be synthesized into DNA because it's for RNA. So you do the same thing. So the second you need a P, it won't work. So you go, oh, I need an A, then I need a C, I need a T, so that won't work. It's going to be 5', 3'. Make sure you switch these around because you have to write 5' to 3'. And the last one, GTP. So you do have guanine, but it's not a dinucleotide, so it won't work. So second you need a G, which would be the fourth one. It won't work, so you're going to do TCA, 5', 3'. So those would be the answers, as long as you put them 5' to 3' first for 5. Basically, you got to figure out what nucleotide one attaches to where.
Okay, so 5 gives a list of proteins that are involved in DNA replication. So this is the list. Boom. All right. So it says which of the above proteins is found in prokaryotes. So above we have DNA gyrase, TUS, DNA A, DNA primase, DNA polymerase 1, DNA polymerase 3 alpha, and those are found in prokaryotes. It says which of the above proteins are found in the mouse, MUS musculus. And we get CDC6, MCM helicase, DNA gyrase, DNA polymerase, DNA primase, DNA polymerase A, and PCNA, and telomerase. C, it says which of the above proteins is involved in chromosome compaction, and that's DNA gyrase. D, it says which of the above proteins is involved in the initiation stage of DNA replication. CDC, MCM, helicase, and DNA A. And lastly, E, which of the above proteins is involved in the termination stage of DNA replication? And that is DNA gyrase and TUS. And that is it for this week.
Thanks.